natural environment that budding yeast evolved to inhabit can be rich and abundant, but it can also be a place of deprivation where essential nutrients are scarce or non-existent. Under such stressful conditions, yeast develop into spores, which are long-lived and durable gametes. Yeast sporulation couples the scrambling of genetic information through meiosis with the packaging of the resulting meiotic nuclei. In my lab in the Department of Molecular Genetics at the University of Toronto, we use the sporulation program to understand fundamental aspects of development. In the course of our study as a graduate student in the lab, Michael Eastwood made a fascinating observation. Using live cell fluorescent microscopy, Mike was following the 4 meiotic products and observed what their fate was during low-carbon sporulation. In a very short period of time, 2 of the 4 meiotic nuclei simply vanish. Now, work in Breck Bayer's lab many years ago found that under reduced carbon conditions, yeast cells execute meiosis normally, but only package 2 out of the 4 meiotic nuclei into spores. They hypothesized that this was an adaptive response that balanced spore number with spore fitness. More recently, research in the labs of Aaron Neiman and Michael Knopp showed that yeast do not package 2 spores under this condition merely because they run out of nutrient to fuel spore construction. Rather, this is a developmentally programmed decision. Up to this point, however, the fate of the meiotic nuclei that did not develop into spores has not been investigated. When we saw that these discarded nuclei vanished rather acutely, we immediately thought of programmed cell death mechanisms. What we describe in our paper is that the unpackaged nuclei are actively destroyed through complementary cell death-related pathways. In the first pathway, the protein components of nuclei are destroyed through an autophagic pathway which most closely resembles mega-autophagy, a much less well-studied autophagic mode. During mega-autophagy, the vacuole becomes permeable, releasing hydrolytic enzymes that consume the cell. Mega-autophagy is an autophagic pathway that has been described in plants and is quite similar to mammalian lysosomal membrane permeabilization. What we found is that following the completion of meiosis, the vacuoles appear to become permeable and then blow up. We next evaluated the other principal component of nuclei, the DNA. Mike found that during programmed nuclear destruction, DNA was fragmented into nucleosomal ladders of approximately 150 base pair periodicity, a hallmark of mammalian apoptosis.
Through the course of studying the cell biology of programmed nuclear destruction, sporulation, we utilized liquid sporulation protocols, which are almost certainly not representative of what yeast would encounter in their natural environment. To ask whether PND is something that they actually utilize, we looked at wild isolates, which normally possess a very robust sporulation program. We examined them as they grow in colonies, probably the best circuit we can find to what they actually do naturally, and found that not only did they sporulate, but they sporulate to a reduced spore number and robustly execute PND. This suggests that PND is indeed probably a very useful and routine aspect of yeast's natural life cycle.
Ultimately, our work reveals a new aspect of the yeast life cycle that involves developmentally programmed convergence of complementary cell death-related pathways. In addition to opening up the studies of programmed cell death mechanisms to the powerful tools of yeast genetics, our work has fascinating implications concerning the evolution of these programmed cell death mechanisms.